What's up, you guys? So in this video, I wanted to discuss dihybrid crosses, as well as the different generations within Mendel's experiments, and kind of connect them all back to Mendel's laws/principles. So if you recall from the last video, and if you haven't watched that video, go watch that now. But that is discussing the principle of dominance, the law of segregation, and the law of independent assortment. So we will see how all of them apply in a dihybrid cross. So looking over, just as a quick recap of Mendel's experiments, Mendel took the P generation, the parental generation, of true breeding pea plants. So they are purebreds. They are homozygous for every trait. So here we have homozygous dominant round seeds and homozygous dominant yellow seeds. I know this isn't exactly yellow, but this is the closest marker that I had to yellow, so bear with me. But we have round yellow seeds, true breeding round yellow seeds, and here we have wrinkled green seeds, true breeding for that trait as well. And when we cross the two, we get all 100% round yellow seeds. And that's because this parent could only give they could only give one R and one Y. So this parent could only give a big R and a big Y, two dominant alleles in that parent's gamete and that parent's sex cell. And here, this parent could only give a little R and a little Y in their respective sex cell. So all of the offspring, all of the resulting children, basically, in the F1 generation, the following generation, are going to be double heterozygotes. So they're going to be big R, little r, big Y, little y. And we call this the F1 generation. F stands for filial. If you take Latin, you would know that filius means son. I believe filia is daughter, but this is the son or daughter generation. And that's the son or daughter generation. That is the first son or daughter generation. So what Mendel then did was he took his F1 offspring and he self-pollinated them. So pea plants have both a male and female part to their flower. So they just took the pollen, which is the male part, from the male part, and just took that pollen and fertilized the female part, and just let them make offspring. And so Mendel counted tons and tons of peas to get a lot of data to kind of go along with the idea that large amounts of data help align with good probability values. But regardless, that's a bit of a tangent. Mendel took this dihybrid, this double heterozygote, crossed it with itself, effectively crossing a double heterozygote with a double heterozygote, and he got the F2 generation. So let's talk about the F2 generation and that particular cross, the F1 cross. So if you recall, the law of independent assortment states that if you have two different genes, and if they're on two different chromosomes, those alleles will segregate independently of each other. So the separation of the R alleles is not going to affect the separation of the Y alleles. So all of these combinations of alleles in the gametes and the sex cells are all equally likely. So this right here is a Punnett Square used for dihybrid crosses, where we're crossing multiple traits, not just yellow and green, but yellow and green we're considering, as well as wrinkled and round. So we're considering two traits, the trait for seed color and the trait for seed shape. So the way we set up a Punnett Square, whether it's a simple Punnett Square or a more complex one, is we always put the possible gametes, the possible sex cells, or the genotypes of the possible sex cells in or on the sides. So here we have big R, little r, big Y, little y. Gametes need to have one of each type of allele. So here we could do a big R, big Y as one possible allele. we could do a big R, little y as another possible gamete. I'm not sure what I said before, but these are both gametes. So I'll put gametes. We could have little r, big Y as another possible gamete. Or finally, we could have little r, little y as the final possible gamete. So these are all of the possible sex cells that the female part of the plant could produce if the female has this genotype, this double heterozygote genotype. Now, if the father also has that genotype, or I should say the father part, or the male part of the plant, also has that genotype, that plant will produce the same gametes, the same sex cells, except they'll be pollen, because it's a plant. But I'm going to draw a little tail like it's the sperm, even though it's pollen. So this isn't entirely accurate, but I'm going to put that there just so we know that that's the male gamete. But it's the same thing. We're big R, big Y, big R, little y, little r, big Y, and little r, little y as our possible gametes. So, now that we've set up the Punnett square, we just need to carry it out. So we have big R, big R, big Y, big Y. So if this pollen grain fertilizes this egg, we will get this genotype in the offspring. If this pollen grain fertilizes this egg, we will get this genotype in the offspring, and so on and so forth. So let me just take a second to write these out. So big R, big Y, little y, big R, little y, big R, little y, sorry, this is a big Y over here. Big R, big Y, little y, big R, little y, little y, big R, big Y, big Y, big R, big Y, little y, big R, nope, little r, big Y, and this part you can honestly just skip through because this is just time-consuming stuff. But little r, big Y, little y, big R, big Y, little y, big R, little y, little y, little r, big Y, little y, and finally, if this sperm fertilizes this egg, or I should say this pollen grain fertilizes this egg, we get 2 little r's and 2 little y's. So that's how we carry out a dihybrid cross. Now, if we were to count these up, if we were to figure out the, we already have the genotypes, if we were to figure out the phenotypes of this particular cross, we would see a very specific ratio. So let me just have my green and technically orange, but we're going to pretend it's yellow. My green and orange markers. So if this offspring is big R, big Y, it's going to be yellow and round. Here, it's also going to be yellow and round. Here, it's also going to be yellow and round, because the dominant allele kind of blocks the recessive allele. So if we see a dominant and a recessive allele present, the dominant will show. That's Mendel's principle of dominance. So here we're seeing the principle of dominance at play. And we're also seeing Mendel's law of independent assortment. And also within Mendel's law of independent assortment, you can also see that the R's, we're only passing on 1 R for each gamete. So that means that the R's segregated and the Y's segregated. So we're seeing all 3 of Mendel's principles / laws in this example. So again, just to carry it out, yellow and round, This one is yellow and round. This one's also, or actually, so this is our first round and green. So let me draw that in here. So we have our first round and green. Back to round and yellow. Now we have our round and green over here too. Round and green. Over here we have another round and yellow. Here we have another round and yellow. Down here we also have a round and yellow. Now here we have a wrinkled, our first wrinkled and yellow. So I'll draw that like this. So we have wrinkled and yellow. Here we have wrinkled and yellow again. So wrinkled and yellow. Here we have round and green. So round and green, and we'll put that there. Here we have wrinkled and yellow again. Wrinkled and yellow. And then as our final offspring genotype, it shows a phenotype of wrinkled and green. So I'll put that up here, wrinkled, and I can make that a little better. Wrinkled and green. So, if you have been counting these, you might notice that there are significantly more round and yellow than anything else. So those are the two dominant traits. So we have 9, if we count them up, 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 round and yellow, so dominant, dominant, when we do a dihybrid cross like this. We have 3, or I should say, so 3 round and green. So 3 of the dominant round and recessive green. We also have 3 of the wrinkled and yellow. So we have the recessive wrinkled and the dominant yellow. And then we have 1 of the wrinkled and green, where both traits are recessive, wrinkled and green. And it's important to note, this is a ratio that you should commit to memory. This 9 to 3 to 3 to 1. We always get that ratio if we're dealing with independent assortment, where the alleles for the genes of interest are on different chromosomes. If we're considering 2 traits like this, and if we do a double heterozygote with a double heterozygote, we're going to see a 9 to 3 to 3 to 1 ratio in the F2 offspring. So just to kind of go back to our original drawing. So the P generation, the parental generation, are purebreds, they're true breeding. They produce all double heterozygotes, or dihybrids, if you will. And then we self-pollinate those double heterozygotes, and we get what we just saw, a 9 to 3 to 3 to 1, 9 dominant dominant, 3 dominant recessive, 3 recessive dominant, and 1 recessive recessive. So that just shows all 3 of Mendel's principles and / laws. And so again, if you haven't seen that video, make sure you check it out. But I hope that was helpful.